Welcome to part one of our chapter 3 lecture in which we're going to be covering cell structure and function. And so we'll do this in two parts, largely just because we want to split it up. It's a little bit big, but we're going to talk about both structure and function together, and that'll help us understand what's going on with our cells. A good place to start is to figure out why do we call them cells in the first place. The gentleman who gave them this name is Robert Hooke, and he was looking under a microscope. It may have been a kind of simple microscope at low magnification, but it's very similar to the light microscopes we might use today. And what he saw were a lot of empty chambers. And so he believed these looked like monks' chambers, which were basically empty rooms, very little as far as furnishings within them. And so he gave these little chambers the name cell. It makes perfect sense when looking at dead cork because, as we already know, there is a cellulose cell wall that's going to surround plant cells. Now, cells actually have stuff inside them, and we know a lot more about that today, so we can kind of flesh out this chapter about our understanding of what cells are and why they aren't just empty chambers. We'll begin here with cell theory, though, to explain the kind of context for cells in our life. Number one, a cell is the basic unit of life. That means that anything smaller than a single cell we wouldn't view as living. And of course, this relates to this idea of number two, that all living organisms are made up of cells. That is, a multicellular organism like us is really, to a great extent, a lot of cells that are working together. And that kind of can become pretty important. If we were to give you, say, a medication, there are probably medications that would maybe target particular cells or target cells in general, but they're going to have an effect by targeting you at the cellular level. Now, our third component here is that new cells arise only from pre-existing cells. And that's kind of an interesting idea. We can see something that relates to that in the upper right-hand corner. As we take a look in the upper right-hand corner, we see milk about ready to be pasteurized. And that is the idea that if we take this milk and we heat it up warm enough and for long enough, we can kill the single-celled organisms that are living inside of that. And then if we place that into a sealed container that no living organisms can get into, we now have a safe food supply. So this idea can be important for food safety. It's also important to point out, though, that at some point in the history of Earth, new cells did arise from something other than pre-existing cells because there would have been a point where there were no cells. We just recognize that that doesn't happen today. Now, the other picture on this page also talks about this idea. We have a cell, it has chromosomes which contain DNA, it can duplicate those, the cell can divide to give rise to new cells, so that's something we're going to want to follow up on later. Another question we might want to answer before we start talking about the specific parts of a cell is, why are they small? In other words, why should we have a multicellular organism with many cells rather than bigger organisms that have large single cells? And there's something here about the relationship between surface and volume. So the basic rule here is that surface area increases more slowly than volume does. So as we take a look at some of these cells over here, we have a single 4-centimeter cube. We could divide this up instead into 8 2-centimeter cubes, or even as small as 64 1-centimeter cubes. So depending on how long they are on each side, if we have cubes, we get more and more individual little cubes in here, but we keep the volume the same. And that's the important thing. Each cube is going to have a surface area, which is height times width times the number of sides. And then as we add more cubes, of course, we have more cubes, each of which has a height, width, and number of sides, and so we're going to get more surface area. But volume is just going to be the total height times the total width times the total length. And as we get that, it doesn't change the volume, the contents on the inside, as we get more and more of these small cells. So you might wonder, though, why does that matter? Well, nutrients and waste travel through the cell membrane, which is the outer surface of a cell, and the cell does stuff with those nutrients or produces those wastes on the inside. So if we start getting more and more inside, and less outside, we have trouble bringing in enough food for that cell to run off of. And we have trouble actually getting rid of all the wastes that are associated with it. So what kind of cells do we have? There are two fundamental different kinds of cells, and these are different kinds of organisms. We have prokaryotic organisms and eukaryotic organisms. Now, as we described life earlier in this semester, we talked about the bacteria, the archaea, and the eukarya as being the three domains of life. The bacteria and the archaea both lack a nucleus, and therefore are what we call prokaryotic cells, where that means before the nucleus. They also lack a lot of other membrane-bound organelles, so while we did say that eukaryotes were more closely related to archaea than either is to bacteria, if we were to look at them, prokaryotic cells are going to be smaller and simpler in appearance than eukaryotic cells, making bacteria and archaea seem somewhat alike. And we see an example of that over in the left side there. Now, of course, eukaryotic cells, number one, have a nucleus, and that's that big ball here we see kind of towards the upper part of this cell. But you'll also notice a lot of membranes inside, including little organelles throughout it which are based off of membranes. And so those are the kind of things we're going to want to talk about since these are our cells. Interestingly, for comparison purposes, there's a virus up top, and viruses tend to be relatively small. Again, we may recognize that a virus might do something like maybe even infect a bacterium, so they will tend to be smaller than the bacteria, which tend to be a great deal smaller than our cells. All right, and this idea takes us to a discussion of how we got eukaryotic cells. Where did they come from? How did this work out? And a big part of that discussion is what we call endosymbiont theory. And we can see over to the right a basic idea of what's going on with the evolution of eukaryotic cells. We, of course, will focus a little bit on animal cells here. But there's an idea that we've got a cell here which could be prokaryotic. It has DNA, as all living organisms do, but it has just a plasma membrane around the outside. There's nothing around the DNA keeping it in the middle of the cell. And so a first step in the evolution of eukaryotic cells would be the evolution of a nucleus. That is some kind of wrapper around the DNA, keeping that separate from other components of the cell. Now, it suggests here for number 2 that this cell would gain what we call an endomembrane system. And that's just adding more membranes and membranous folds which are connected to the outside of the nucleus. And we can see some of those around the outside of the nucleus. These are going to be organelles like the endoplasmic reticulum and Golgi apparatus that we'll talk about later. And then here's the big step that relates to endosymbiont theory. We've got this idea of gaining mitochondria, these additional organelles that we have in our cells, which it shows here as being what we call an aerobic bacterium. And so that mitochondrion is thought to have come from a separately living bacterium which is capable of using oxygen to use glucose to generate ATP in a very efficient manner. And we'll talk about that later on. So this idea here means that this cell, this eukaryotic cell, by taking in this bacterium and living with it, is going to become more efficient. That is, it's going to be better at metabolizing sugars to produce ATP and can become a more successful organism. But really, it's two organisms living together. That's the very meaning of endosymbiont theory. Endo is inside, and symbiont is to have this long-term association. Now, obviously you're wondering, boy, is there any evidence for this? It sounds kind of like, a far-fetched story. And the reality is it's actually very well supported by evidence. A couple of the main pieces of this are the fact that the mitochondrion has extra membrane around the outside of it, so it's a double-membraned organelle, and that it has its own DNA. In fact, that DNA, rather than looking like a chunk of DNA from the nucleus, that maybe is now just in an organelle that is replicating itself, rather that DNA appears to be bacterial type DNA. So it has more similarities to bacteria. We also see the same thing happening with chloroplasts. Now, of course, our cells didn't gain those, but the cells that led to plants did gain those, and we see exactly the same kind of evidence suggesting that not only is endosymbiont theory the history that we have for eukaryotic cells, and that we can explain the complexity of eukaryotic cells in part because they have kind of gone into these associations with bacterial cells, but also that this thing has happened more than once. And so that's kind of an interesting fact about the history of life. All right, so one of the things we need to understand very well to understand cellular behavior and physiology within humans is the plasma membrane. That is the outer wrapper of a cell. So, of course, all cells, both eukaryotic and prokaryotic, are surrounded by a plasma membrane. And we explain this plasma membrane by something we call the fluid mosaic model. And so that fluid mosaic model basically tells us that most of our plasma membrane is made up of these little guys in here, a little ball and two tails, forming what we call a bilayer. That is, there are two layers of it, where the tails face in towards the middle and the heads face towards the outside. So those are our phospholipids. And within the phospholipids, there are proteins that are embedded in it and can move around. That's the fluid idea, that these molecules that form the plasma membrane are not rigid and rigidly attached to each other, but rather can flow. But it's a mosaic in that these big chunks, these proteins that are embedded within the phospholipid bilayer, are capable of moving from one spot to another, or we can add more to it so that we can change the relative ratio of proteins that are embedded within this phospholipid bilayer. Okay, so the things we need to know about the phospholipid bilayer are, again, that it has heads and tails, that the heads, composed of phosphate, are polar, and therefore they can be adjacent to water, whether it's outside the cell or inside the cell. But the tails are fatty acid tails, so they are nonpolar, they are hydrophobic, and that's why they face each other. Now, this idea that these tails are hydrophobic is also going to give us one of our basic properties. As you can see in this picture, some things are moving through the phospholipid bilayer, and others have to move through protein channels. Okay, so basically, polar items are going to need a protein channel to get through, and non-charged or non-polar molecules may be able to move through the phospholipid bilayer. All right, so how do we get what we refer to as selective permeability, this kind of control over what goes in and out of the cell? Now, the short answer to this is that there are going to be two fundamental ways in which stuff can cross the plasma membrane. If we want to break this up easily, either things can move passively, that means without energy, or actively, and that would mean with energy. And so when we think about passive ways in which things can get across the membrane, we start with this concept called diffusion. Diffusion does not have to take place across a membrane, but it can. It is simply the random movement of molecules from a high concentration to a low concentration, such that the difference in concentrations will disappear as this diffusion takes place. And we can see that down below. As we look down here, we've got a purple particle on two sides of a membrane. And what we're seeing here is that we have more of them on the outside initially and less of them on the inside. Although we're going to be getting just random movement of molecules, we're going to see a net movement of things towards the inside, and that's eventually going to set us up with what we call an equilibrium condition. So that equilibrium condition is going to mean that now we've got water and purple particles evenly distributed on both sides of this membrane. Now, this idea of becoming equally distributed really does depend a great deal on whether things can or cannot cross the membrane, so that's something we want to keep in mind. So to sum up diffusion, molecules move down a concentration gradient. That means from high to low. If we've got a lot of purple particles outside, they're going to move inside. If we've got a lot of water outside, it's going to move inside. Okay, and this all kind of depends on concentration gradient and the capacity of these things to move across the membrane. Diffusion is particularly important for things like nonpolar molecules. And as we remember, oxygen gas, because the two oxygens are the same, would be a nonpolar molecule, or that is that the oxygens are bound together in a nonpolar covalent bond. So when we think about the idea of air being something that we would breathe in, and that we have blood flowing to our lungs to pick up oxygen, when oxygen is in our lungs and needs to get into our blood, it can simply cross the plasma membranes of our cells going from that high concentration in what we call the alveoli of our lungs across the membranes into that area with a lower concentration inside of our bloodstream. Now, of course, that's something we'll talk about again later. Now, there are some special cases of diffusion, and osmosis is a good example of that. Osmosis is essentially the diffusion of water. So you can see here it's defined as the net movement of water across a semi-permeable membrane. And when we think about this, water can move weakly across the phospholipid bilayer, but water is polar, and there's going to be some restriction to movement. But there are protein channels called aquaporins, which allow for the flow of water across those. So when water has this potential to move from one side to another, we can use the term tonicity to compare two different areas, outside and inside of a cell, for example. So tonicity is the relative measure of the difference in solute concentration between two areas separated by this semi-permeable membrane. Now, usually we're going to be talking about this cell membrane here, but let's say if you needed to undergo, say, dialysis treatment because of kidney problems, it would be the same basic setup. So the way this works is we've got a few things going on. We have a solute, that's a dissolved substance, and it's going to be in a solvent. For our purposes, that will generally be water, and that creates a solution. So a solution can either have a lot of solvent, or it can have a little solvent, or a little solute. And if it has more solute, there's less room for water because there's more of whatever that dissolved substance is. And so we can think here about some different scenarios, and we have some different terms we'll need to use. Now, this whole idea is very important for physiology, and as you can see, this image here addresses what happens to blood cells in different environments. We'll start with an isotonic environment. Iso is basically a word that means the same. So we have the same tonicity inside the cell and outside the cell. And as a result of that, there is what we call no net movement. That is, there's always going to be random movement. We can never say that there will be no movement unless we have no movement of molecules at all, which occurs only at absolute zero. So in this case, we should have water from this beaker moving into the blood cells, but an equal amount of water will move from the blood cells out into the beaker, and so we're not going to see any kind of change. Now, in a hypertonic solution, we have a higher solute concentration than another solution. So one area has a higher concentration. And so as we think about hypertonic, which we see in the far right, we have the equivalent basically of what we might refer to as salty water. Now, it could be incredibly sugary. There's some kind of dissolved substance in there, and there's more dissolved substance in the water than in the cells. And you'll notice here that primarily we have an arrow going from the cell out. That is, the cell is losing water and it's actually shrinking. And we can see what red blood cells might look like when they have shrunk in an actual solution like this. So water is pulled out of the cells into the environment. Water will still enter the cells, but not nearly as much as is exiting. Now, our middle picture shows us hypotonic. So we're sticking cells in a hypotonic environment, one that has a lower solute concentration. For our purposes, that's a very watery environment. In fact, you can't get more hypotonic than, say, pure water that has nothing dissolved in it at all. And what you can see here is, since the water content is higher in the water, that is, it has less dissolved substance in the cells, water will move from the environment into the cells, and they are swelling and getting bigger than they're supposed to be. Of course, if we use something like distilled water, not only will water start moving into the cells and will they get bigger, but because we can't reach an equilibrium, in other words, we can't make pure water have the same amount of dissolved substance as something that does have dissolved substance, these cells might actually explode on us, and then we'll obviously lose the contents. Okay, so what if I have something that won't work its way through the plasma membrane? That is, it's maybe polar, maybe it's kind of a bigger molecule, but I still have a concentration gradient, meaning I have a lot of it piled up in one place and little of it somewhere else. In the case of the picture we see to the right, looking at this green substance, it looks like it could be something like glucose. I've got a lot of it on the outside and less on the inside. And so as we're thinking about this, facilitated transport is diffusion. That is, it occurs down a concentration gradient, but it's for something that can't make it across the membrane by itself. And so this protein that we see in here is what is facilitating this diffusion or this transport. It's allowing the glucose to move from the outside to the inside when it can't do that through the phospholipid bilayer, but we still don't have to use energy because it's flowing from high concentration to low concentration. So importantly, facilitated transport requires a protein, but it does not require energy. And we want to contrast that with this other protein down here. This happens to be what we call a sodium-potassium pump. And this sodium-potassium pump is going to do exactly what it says, and that is to pump stuff. And pumping requires energy. But it allows us to do something we haven't yet been able to do, and that is to go up a concentration gradient. That is to go from a low concentration to pump something into an area of high concentration so that we can essentially stockpile a substance in a particular location. So active transport is different in that it requires energy. And the action we see here is that we're going to use the energy in ATP, adenosine triphosphate, to change the shape of this protein, which binds in some places to sodium and others to potassium. So it's going to be able to pick up sodium and pump it to the outside of the cell, which will become very high in sodium relative to the inside. And then that same protein is going to pick up potassium and carry that to the inside so that we stockpile potassium on the inside. And so what we've done is set up a scenario where we have lots of sodium outside, lots of potassium inside, and we'll have the possibility to use this to do something very, very interesting. And we can see a little bit of that here. So what we're going to talk about is nerve transmission, and we can see in this case that we have some changes that take place. We're trying to compare the inside and the outside of the plasma membrane of an axon, that is a nerve cell that's going to, or a neuron, a component of a neuron, which is a cell that transports signals from one location to another. Basically, they're the things that make us understand that we've received stimuli, so we can sense things like touch or sight, or that send signals out to our muscles so that we can move. And so you can see here we start off with this potential of -70. We have a negative charge on the inside and a positive on the outside. We have a lot of sodium built up on the outside and a lot of potassium on the inside. And we have what are called gated channels. You can see that they are gated sodium channels or gated potassium channels. So our sodium-potassium pump has set up concentration gradients, but we have proteins in here, these channels, which are going to allow for facilitated transport. And what you can see here is that we are changing things across the membrane. We are making it more relatively positive on the inside and negative on the outside as we start to allow the flow of sodium ions. And you can see here we end up with definitely a different charge across that membrane. This positive charge moves its way down the axon, and as it does that, we're eventually going to have to reset this whole system, which will begin with the opening of potassium channels. And that's going to allow from the inside some positive ions to flow to the outside, and we're going to go back to our initial potential we had here at -70, and you can see again we're going to be minus on the inside and positive on the outside. Now all that's left for us to be able to send another nerve transmission is for those sodium-potassium pumps we saw earlier to reset the sodium and potassium. So we'll talk about this in much more detail in the nervous system chapter, but I want to set this up now so that we understand that channels, whether they may be for active transport or if they are for facilitated transport, are going to be important in basic body functions. All right, and we're going to end here with how do we get bigger things into or out of the cell. And we're going to start with this idea of endocytosis. Endocytosis is where we invaginate, and we can see that up top here. We form this little indentation of our plasma membrane, and then we bring the tops over so we can pinch off what we call a vesicle and bring a food item inside the cell. Now, later we're actually going to have another vesicle bind to this, and add some enzymes which are going to help us digest whatever that food particle is, to break down some molecules into smaller pieces that we can use. But we first have to take those things in. So this particular case is phagocytosis. It's used for large objects. And we see up here a white blood cell, a neutrophil, which happens to be known as being a phagocyte, which is going to eat this little yeast particle here, this yeast cell, and so it's going to wrap around it, engulf it, and then destroy the inner contents or break them down. So we refer to phagocytosis as cell eating. And this takes place in more than one way, but this action of white blood cells to deal with pathogens, or even to break down cells that have worn out in our body, is one of the important ways in which phagocytosis takes place. Now we do see in the diagram here pinocytosis and receptor-mediated endocytosis as two other forms of endocytosis we're not going to focus on, but you can see that pinocytosis is called cell drinking, that we're just basically taking a swallow of whatever's in the outside environment so that we can take large amounts of things in, but it's not particular stuff, it's just what's out there. And in receptor-mediated endocytosis, we are taking in particular particles. That is, we have receptor proteins which bind to something in particular, which line this vesicle that we're going to pull in, so that we are pulling in more of a particular substance than we would in a normal pinocytosis. Okay, now if we have cell eating, we need to have the opposite of it as well, and we call that exocytosis. So in that case, you can take this phagocytosis up top and kind of put it in reverse. In this case, we would take a vesicle like this, and we would bring it to the edge of our cell and open it up and be able to have this substance, maybe it's a mucus or some other cell product that we've made that we need to get rid of, we're going to put that out of the cell. So again, if we have a cell that is glandular, producing something like mucus or hormones, exocytosis may be the mechanism by which we can get rid of those and then have them distributed to the area of the body in which they're going to be used.